All right. Well, let's get started. My name's Kyle Bittinger. This is the second of two workshops that I'm doing this month on kind of background for microbiome studies. I direct the analytical group for the Penn Chop microbiome program. So I have two bioinformaticians that I work with, Jaylon and Chunyu. And I'm a new assistant professor in gastroenterology at CHOP. Okay, so last time we talked about bacterial taxonomy and how one might take DNA sequences and turn that into information about bacterial species or bacterial families, and how different ways that you might manage the uncertainty about what to call a bacterium, or how to cluster sequences into species-like groups. So for today, we're going to assume that we've done all that perfectly. We have great-looking clusters. We know what everything is called. And today, we're going to move beyond just getting names and sequence clusters, and we're going to talk about how to analyze the content of samples and compare samples to each other. So this is the outline for my talk today. So I'll give kind of a general overview of ecological diversity and talk about why we're so obsessed with that idea. And then the specifics in the lecture will cover alpha diversity for about half the time and beta diversity for about half the time. And I'll tell you all about what they mean, what their history is, and how those things are measured. And then I made another application for today where we can try out, so it has like 2 samples, and each sample can have 6 bacterial species. And as you adjust the levels of the bacteria, it will adjust the different diversities, and the dissimilarity between samples will change. And so you'll get to see how all these different measures are responding to changes in the bacterial proportions. Aurea suggested last time when I gave my talk on taxonomy that I start with a slide of the 16S gene, and so I'll do that today. This is the gene that is sequenced from bacteria to kind of survey bacterial communities. The reason that this gene is used is because it's very highly conserved in bacteria, and one can make PCR primers that will target specific regions of this gene, and those primers can be made such that they're universal. They'll hit the gene for almost all bacteria, and they'll never match to any eukaryotes. So you can capture all or almost all bacteria, get everything from the domain of life where you want, nothing from domains of life that you don't want. And then between the primer sets, this gene has short regions of where it's more highly variable. And so those short regions of variability can be used as a fingerprint for the bacteria. And the gene is arranged wonderfully so that these regions of variation are like almost evenly spaced, and the intervals at which they're spaced correspond really well to changes in sequencing technology. It's an RNA gene, forms part of the bacterial ribosome, so there's no codon structure in the DNA. And I think that's it. Yeah. And so one of the, this is the really important thing, is that this is the gene that's used for bacterial phylogeny. So when you sequence this gene, you get phylogenetic information about the bacteria in your community for free. You're already sequencing that thing that they use to make the phylogenetic trees just as your fingerprint. So that's your bonus information. Okay, so why do we care about diversity? Well, we're concerned in the land of the microbiome, we're concerned with studying communities of microorganisms. And to start with that, we'd like to say who's there, how many of them are there, and how they're arranged in space and in time. And there was this paper that came out, Jessica Mark Welch and had published this in PNAS last year. It's microscopy of bacteria in plaque. And what these pictures illustrate wonderfully is that bacteria are not randomly arranged in space. So they have very specific, seemingly, interactions with each other. They're growing. Their pattern of growth in space is not random. Their locations in space are not random. And so when we talk about diversity in general, we'd like to quantify how is this region different from a neighboring region. Are we changing a lot as we go this way, or are we changing a lot as we go this way? And that's what diversity is getting at. There is more to the microbiome than just studying diversity, but diversity is your starting point. Here's just another picture from that paper showing the arrangement of bacteria kind of growing on the tips of this, this cauliflower-like structure. And again, this like showing the backbone of Corynebacterium supporting a variety of other bacteria towards the edges of the structure. So other kind of motivations for species counting, tracking diversity. This is a classic ecology experiment by Simberloff and Wilson. 1969, they were doing this experiment on tiny islands, I think off the Florida Keys. And what they did is they went to each island and they gassed all the living organisms on the island and killed everything. And then they waited to see what would come back. And it's kind of crazy. It sounds kind of like an evil experiment, but they were mostly killing butterflies and millipedes. So, you know, it wasn't like they were taking out small animals or something like that. But anyway, what you see, so this is the life on the island before the experiment. This is what was on the island after Simberloff and Wilson came. And there's only two things left. And so what you'd like to know coming out of an experiment like this is, well, after 360 days, did life on that island look basically like it did before we intervened? And how fast did organisms come back? And did organisms come back in waves? Were there some founders that maybe went away, and then there were some organisms in the middle, and then the organisms that were there at the end, when did they arrive? What part of the process? These are all questions of diversity and species counting, and it's where we start to understand experiments like this. So this seems like a totally evil experiment, but we do the same thing with bacteria all the time. We have no moral opposition to killing bacteria, and so we give antibiotics to mice and clear out bacteria in experiments all the time. So this is an experiment that we did in Rick Bushman's lab. Serena Doliv was the graduate student who did this. She gave a cocktail of 4 antibiotics to mice, got rid of all the bacteria. In fact, during the antibiotics segment of the experiment, we sequenced the bacterial DNA from the feces and got DNA from mouse food. And so what this illustrates really nicely is that over the course of 18 days following that experiment, you have these waves of organisms that come and go. So in the beginning, proteobacteria are really dominant, and then you kind of come into one type of Firmicutes, which on day 28, eventually gives way to a mixture of Clostridia and Bacteroidetes, which is kind of in the kind of the same arrangement that you might expect to see in a normal mouse. So we want to characterize like how much is the community changing as time goes on, when does it achieve its final state, how close to the beginning did the final state look. So we're doing these type of experiments, and diversity is our first step. Okay, to kind of give a general definition of ecological diversity, I think about diversity as anything that involves species counting. So if you're counting organisms in species and you're tallying those up somehow, you're in the realm of diversity. And that'll be kind of my working definition. So you can count simply the total number of species present with at least one organism in a sample, or you can weight the diversity by the abundance of those organisms. So, and we'll talk about several ways to do that. This is the classic species area curve for birds. So when you have a larger island, the island can hold more types of birds, and when you have a tiny island, you can only have fewer types of birds on the tiny island. So classic result from ecology and an example of looking at richness and plotting that against some ecological gradient. Okay, so richness means the total number of species observed. We can't say species when we're talking about bacteria, so I'll say OTUs. For the purposes of this talk, I assume that we've made great sequence clusters. We've solved that problem. So it so happens that when you count more organisms, you tend to see more types of organisms. And so if you have a bunch of different samples and you've counted different numbers of individuals in each sample, how do you put them all on even footing? There are two basic approaches to this. The first strategy is a strategy of rarefaction, or removing some of the data. And this is the safe strategy, generally. So you work backwards. You say, well, what if I had only counted 100 organisms? How many species would I have seen at that level of sampling? If you're just concerned with the total number of species, there are equations that you can just solve algebraically that'll give you the expected value and the standard deviation for the number of species at any sequencing depth. Okay, second approach is to extrapolate. Ann Chow basically introduced these methods to you take your abundance distribution of individuals that you've counted so far, and you look at all the species that are rare, so species that you've just counted one or two individuals for. And then you expect, if you're seeing a lot of species where you've only counted one individual, you might expect that as you continue to count, you'll be uncovering more and more species. And so her index tries to estimate what the total number will be based on your information so far. And this is something that works better for macroscopic experiments, but I don't suggest it for sequencing experiments. The reason I suggest staying away from the extrapolation methods for sequencing experiments is because new species are a mixture of sequencing artifacts and legitimate species. And it's often tough to disentangle those two varieties of OTUs. So this was a result from Mitch Sogan's lab in 2006, reporting on the rare biosphere, or the variety of rare species that they were observing in ocean water. It turns out that a lot of this was sequencing error, and this really caused people in the field, or motivated people in the field, to come up with methods for identifying and correcting sequencing errors. So it was hugely beneficial for the field, even though many of those species weren't authentic. Okay, going beyond just simple counting of number of species, we can try to quantify diversity by the abundance of species. So let's say we've collected a sample and we have 100 organisms from one species, and then we have 3 other species, but we've only collected 5 organisms or 3 organisms or 2 organisms from the other 3. So we'd like to have some way of saying this one species is very prominent in this sample. It's basically one species with a few extras. So the 2 most popular ways of quantifying that are the Shannon index and Simpson's index. All right. Shannon's index ties into this whole history of information theory, and it's great to read about someday when you have some free time. We're not going to concern ourselves with that today. For our purposes, Shannon's index is calculated by p log p. So you take your proportions and you multiply them by their log, and then logs are always negative for proportions, so we put a negative sign out front so we don't get a negative number. So that's how you get it. What does it mean? It's at a maximum when all the proportions are even. So if you have 4 organisms, if they're all at 25%, then you've maximized this. If you have only 1 organism, this goes to 0. Okay, the alternative index is Simpson's index. So here, instead of taking p log p and adding those all up, you just take p squared and add it all up. And the good thing about Simpson's index is that it corresponds to a probability. So you're quantifying the probability if I selected 2 individuals at random that they would come from the same species. So that's kind of cool. Something to watch out for with Simpson's index is that the terminology is different between people who use the word. So sometimes you'll get this lambda, which gets smaller as you get more species. So it's kind of like the opposite of a diversity. So sometimes people use 1 minus lambda, and sometimes they use 1 over lambda. All of these have slightly different interpretations, but it's something to check when you are using this as an index. All right, now phylogenetic diversity. So we have the phylogenetic composition of our community. We've gotten that information for free, so we might as well use that to quantify our diversity. I think this is best illustrated by extreme examples. So here's what we do. We draw a phylogenetic tree. We put sequences from the red and blue sample on the tips of that tree. And then what we're going to do is color branches that go to one of the red sequences. So these branches on the end all get colored because they're all pointing directly at a red sequence, and then any higher branch that's pointing at a branch that eventually leads to red also gets colored. And we basically take the length of all these segments and divide it by the length of all the branches in the tree, and that's our phylogenetic diversity. So if you have bacteria that span the whole tree, then you're very diverse. If you have bacteria that are basically only from one genus, then you are not very diverse. So that's how it works. And the great thing about phylogenetic measures when you're dealing with sequence data is that the sequencing errors often don't mess up the phylogeny very much. So you're close to the right answer on the phylogenetic tree. That's about as good as you can get. And so if you have sequencing error, it doesn't add a lot to a diversity that's focused on phylogenetics. Okay, and that covers our alpha diversities. So richness, just counting species, weight by abundance, or use the phylogenetic information. We're going to go through the same pattern of stuff with beta diversity. So beta diversity started with Roger Whitacre. He is he's walking up and down mountains in like Oregon or Northern California or somewhere great like that, counting pine trees, and he wants some way to quantify how the trees are changing as he's walking up and down the mountains. This is where the motivation for beta diversity comes from. The reason that it's thought of as a diversity is because you're still counting species. You're still counting individuals in categories. But with beta diversity, you're counting the common species between two samples rather than just the species in one sample. So for numerical reasons, we usually only use beta diversities that describe two samples. And we usually cast these beta diversities as distances between two samples rather than similarities. And so this is an example from one of Whitacre's really early papers. And so he is trying to see here how sample similarity is decreasing as he walks down the mountain. And this is, so he originally is collecting data like this, where you see abundances of different trees going up and down. And so he wants some method to numerically kind of quantify this and be able to do statistics on it. So beta diversity was born. Same pattern as alpha diversity. We'll think first about presence and absence. So we're just counting number of common species, number of species that are not common. All right, two popular ways to do this counting, and almost every way that you can count common species by presence absence, you can frame in this this formalism of A, B, and C. So here's my two samples. They have a bunch of species within them. We're going to classify all those species as being either only in sample 1, only in sample 2, or in both samples. So the Jacquard index, and I'm showing these as similarities, the Jacquard index says what's my fraction of common species out of the total. And I think that's the easiest to explain and to think about. A related way of quantifying that is Sorensen's or Dice's index, where you say, okay, if I have one of these two samples, what's the average percentage of species in either sample that's shared? Gets at the same idea, it just quantifies it in a slightly different way. This one gets a star because it has slightly better mathematical properties. So if you don't care between them, you should probably pick Jacquard's index. And notice about this, so there's a bunch of other species D that don't appear in either one of these samples. But these distances only count the species that appear in sample 1 or sample 2. If you start counting species that don't appear in either sample, you run into weird kind of philosophical problems. Like as I add samples to my data set, I'm changing all the distances. Can samples be more similar to each other because they're both missing the same species? Will that answer change as I discover new species in completely unrelated samples? To avoid those kinds of issues, the types of measures that we use in beta diversity only consider for any two samples the species that we saw in those two samples. Okay, and then again, we want to try to take into account the abundance of species, not just whether they were there or not. Okay, and the most popular way to do that is the Bray-Curtis distance. So if you were going to think about the simplest way to account for differences in abundance, you might come up with this. So I just take all the abundances, all the proportions between the two samples and subtract them, and take their absolute value and basically add that up. So it's the sum of the differences in proportions between the two samples. And right, I'll point you to the, I'm going to try to keep it on my slides where you can go for additional information on these things, because there's like a world of measures, and I don't have time to explain them all. But if you had to pick one for one measure for abundance-weighted similarity between samples, you might as well pick this one. It's simple. It has reasonably good properties. And this is an example from Bray and Curtis's paper in 1957. It's a beautiful graphic. We see like, so we're using a scatter plot, so we're fitting a lot of information into the plotting space. They're using the size of points to describe total abundance of different types of flowers in the sample. We're using faceting to pull out different types of flowers in different subplots. And you can, if you look at this for 1 minute, you can begin to see all types of patterns in here. One thing I love about this paper is that they got so excited about making their plots that in the last figure of the paper is a photograph of a three-dimensional ball and stick model of this data made with wood and wires. It's totally beautiful. But back then you really had to work for your 3D plots. So you might think, well, we already have a way to think about distance between things, like distance between objects in this room, so why don't we just use that? Why do we get into all these complicated distances? And there's good reason to avoid our usual distance. Problem number one is that it has this double zero problem. So things can be more similar by virtue of missing the same types of species. And we'd like to not consider double absences in influencing the distance. Problem number two is that when we use this type of distance with proportions, it scales all weird. So this is a plot. This is a bit complicated, but these people in this article about compositions made this plot such that they're changing some proportions, and they argue that if you change the proportions as they're doing it, you should get a straight line. So the distance should increase linearly as they're changing these proportions. And they've plotted 4 different distance measures, and Euclidean distance is like the worst. And all the other ones might be a little weird, but at least they're going in the right direction. I do have to say, in practice, we're not usually working in this area. So we haven't hit, typically in our data sets, the saturation limit, but there's this argument that it's kind of a bad distance in how it scales with proportions. Good news is that if you log the proportions, it behaves really well. So that's an option for people. So right, the blue line here is the Euclidean distance after you just take the log proportions, and then magically everything works out. Okay, so we know how to do abundance-weighted distances. We know how to do distances with presence-absence. Let's put the phylogenetic information in. I'm not citing Kathy Luzapone's paper, but this is from her original paper on unifrac. Works the same way as Faith's phylogenetic distance. So we're going to draw a tree. This time it's got red sequences and blue sequences in it, and the red sequences are all from Bacteroidetes, and the blue sequences are all from the Firmicutes. And so these samples are kind of maximally different in a phylogenetic sense. Meanwhile, I've collected these other 2 samples, red and blue, and the sequences are all mixed up. So like some of the blue is in Bacteroidetes, some of the red's in Bacteroidetes, and likewise with the Firmicutes. And so these have a very small distance phylogenetically because the species are all mixed up between the samples. And these types of distances are widely used in microbiome studies, kind of for a few reasons. they do work against sequencing error really well for reasons that I mentioned earlier. And also they seem to capture expected differences, and these distances kind of seem to correspond well to differences in environments for bacteria. So there is some of the justification for using these is just because, well, they seem to work really well, and they give us answers that are like the answers we expect. I mean, I guess you could make a bunch of arguments for why that is, but if you really get down to the core of it, like the theoretical justification is basically that. And these come in two flavors, so you can either count these as presence or absence, in which case you're basically doing Jacquard distance between the samples, but then you weight it with the phylogenetic tree. Or you can use the weighted version, in which case you're basically doing Bray-Curtis distance, so you're looking at the differences in proportions, and then you add the phylogenetic weighting. And then you get plots like this. So this is showing 3 body sites, so stool, mouth, and nose, and the fecal material is much different, as you might expect. So how do we get from distances to a plot like this? The practice of making a plot in 2 dimensions from data is called ordination. So our goal here is that we have a distance matrix, that's our starting point, and then we want to get to a plot. That's our output. The 2 primary ways that people attack this are metric multidimensional scaling and non-metric multidimensional scaling. So the metric version is called principal coordinates. And what the method is doing is it's taking that distance matrix and then it's making a space where you've put points in the space where their distance, their Euclidean distance, corresponds to this distance matrix. The problem is that the space has to have like 57 dimensions to get those distances to be right. And so part of this method is picking out the first, arranging the dimensions of that space so that all of my my different directions are perpendicular, but the first axis captures as much of this distance matrix as I can, and then my second axis and third axis capture less and less. But this is what's important in principal coordinates analysis is that the axes are perpendicular to each other. That's what the method won't give up on that. It'll always give you axes that are perpendicular. That's nice, and it'll always give you a space where the distances, the Euclidean distances, are exactly those in your distance matrix. Sometimes the space has to have a black hole to get the distances to work out, but we can do that. It's no problem. The alternative method, non-metric multidimensional scaling, here it says, this method is like, well, I know I want to make a plot that's on a piece of paper, so I only have 2 dimensions to work with. And I'm going to give up on making those perpendicular to each other. And I'm going to give up on representing the distances exactly, but I'm going to try to get the short distances to be shorter on the paper, and the long distances should be longer on the paper. So it relaxes a lot of these guarantees that you get with principal coordinates, but it tries to get the general gist of the distances onto the plot. And so I've taken this distance matrix just as an example. I generated random numbers to make the matrix, so the patterns aren't especially startling, but you can see things like samples B and F are the closest in this distance matrix. They're the most yellow between them. And if you look at the two plots, they aren't especially closer to each other than any other samples. So, which just points to the fact that these plots are trying to balance a whole bunch of different distances to make the final plot. But you do have these these like H, D, and I should be a little bit out there. So the principal coordinates gets D and I off from the main section of samples, and then the non-metric multidimensional scaling does a little bit better and puts I, D, and H all in the periphery of our sample set. They have a different value for doing different things, the two methods. We have a bunch of distances. How do we do statistics with that? So I have a plot of these samples. I see the nasal samples are up here, the oral samples are down here. Are these samples different from these samples? And so how do we test that statistically? All right, the two most widely used approaches are Perminov A. So this was established in 2001 by Marty Anderson. In this approach, the idea was that we want to do an ANOVA to test for differences between groups. And to do an ANOVA, we need this column right here, the sum of squares. So if I had normal data, if I had typical data, like I collected a bunch of numbers from two different groups. In one group I saw numbers like 10.5, 11.5, 13.0. Another group I saw numbers like 5.0, 6.1, stuff like that. Then if I wanted to do an ANOVA on that, then I would have to get the sum of squares difference from the mean. So you have this one column for sum of squares. So this is the difference from the grand mean. This first column for grazers, this is the difference from the group means. So this is your within group differences. This is whatever's left over. That's your between group differences. And then this is the total variation in the data set. Once you have this column and you know your degrees of freedom, then everything else is mechanical. So you just carry out the ANOVA like they taught you in Stats 101. The issue is getting the sums of squares. And what we'd like is to have some kind of mean point for each group in our distances, and then we'd like to take these distances to the mean and get our sum of squares. This is what we want to sum and square and sum. But we don't have this. We don't know where the middle is. We have this. So we have the distances between all the points. And what Anderson shows us in this article is that you can, if you have this, you can get this by an algebraic formula. And you never have to find out where this is. You can get the sum of all these squares just from these point-to-point distances. It's brilliant. And then once you've done that, then you have your sums of squares, and then you can just do an ANOVA like everyone else does. And anything that you can do in an ANOVA type analysis, which is much of statistics, you can carry out in this Perminov A. It's simply converting your distances into a more traditional method that's very well established. That's why I like that approach. AMOVA is a different approach. It's a non-parametric test of distances. It's roughly doing the same thing. It's looking for a difference between groups, but it doesn't have this correspondence to classical ANOVA. So if you're doing like a complicated design, it's not clear how to put your experiment into the AMOVA framework. It's fine if you have a two-group comparison that you're doing. Works perfectly well. And then, sorry, I don't have the reference for this Meerkat software, but this is a newer approach, and basically we're flipping the question. We'll put, instead of having the distances on the left side of our equation and having all of our explanatory variables on the right side, we pick out one of our explanatory variables, we put the groups on the left side of the equation, and we put the distances on the right, and then we try to use the distances to explain the groups. So we're like flipping our equation. And this this ties in with a whole bunch of fancy mathematical methods in regression. And so this is a really rich way forward also for testing. One thing that we can do conveniently, because we have the distances between samples, it's very easy to check if one group is more spread out than another group. And so there's a test for that. Often in disease states, people will be dominated by one bug or another. And so you'll see in the disease population that they'll be more dispersed throughout, be more different from each other just because they're infected with different bacteria. Okay. The number of individuals you count affects your alpha diversity. It also affects your beta diversity. If I have 2 samples and I didn't count that many individuals for the first sample, then it doesn't have that many ways to be different from the second sample. As I continue to count, the samples have an opportunity to be different in more ways. And so this is an issue, I think, if generally if you have a nice distribution of reads per sample and it's not lopsided between your 2 groups, it rarely affects the outcome of your test. But if you want to check on this, there are, again, 2 approaches. So one is to subsample everything in your study to the same sequencing depth and then run the test. Issue there is that you have to pick that level of scaling back your data. And then if you do that randomly multiple times, what if you get different answers from your test? So you can do that. You can take that approach. It has some inconveniences for those reasons. Another approach is to include the sampling depth in the statistical model. So you can actually put the sampling depth in your ANOVA as the first term, and you fit the sampling depth first, and then you try to see if there are differences between groups. And that also works reasonably well. Neither approach completely solves the problem. It remains an open question. Okay, and then the final thing that I have for today is, okay, you're doing this new experiment. You know you're going to be doing these tests between groups to see if there's a difference in bacterial populations. How do you power that? So Brendan Kelly wrote some software to address exactly that question. His software tries to look at other similar samples that people have collected in history, and then it does some simulations on the actual bacterial populations to simulate specified effect size. And then it can tell you what your power is. It actually simulates the samples and then does the test a bunch of times. So that's great. If you want an easy way to do the same thing, you can look up the formula for a Ttest in your statistics textbook. This is how you do it in R. This is a built-in function. You have to give it the power. So just use 0.8. And you give it the number of samples in each group, and then it tells you what your effect size is. So this is the difference in means in terms of the number of standard deviations. So this is like, I can detect, with 10 samples in each group, I can detect 1.32 standard deviations difference in means. And the reason that this kind of works is because we're doing what basically amounts to a Ttest with this Perminov A method. So each group has some standard deviation. We have a difference in the mean samples, and we can phrase this difference in means in terms of the number of standard deviations. And so for a really rough approach, that works just fine. All right, this is the book. Further reading, go here. It's a fantastically thick book. These are the people in the Penn Shop Microbiome program. Chunyu and Jaylon are bioinformaticians at large, so they'll work on a bunch of different projects. Yua, we work is a statistician, and we work with her on one of our big projects, and then we have a full staff at the sequencing lab who can help with experimental considerations. And thank you to everyone. And then I'll remind you to please fill out a survey. It's okay if you don't like the lecture, but I need it back. And I made a demo for today. People at the last lecture used up all my free account on shinyapps.io, so I had to put this up in the microbiology department at Penn. So there you go. And thanks everyone for coming.